I look forward to the opportunity to learn more about the Innovine project. It's a great pleasure to be here, see so many people I know, look forward to meeting people I don't know. And this is a nice opportunity for me to talk to you a little bit about a project that had some similarities with Innovine, but yet some very big differences too. What I'd like to do is tell you about a project we called the VitisGen project. We all have to have our abbreviations. Innovine, well, we have VitisGen. VitisGen was a 5-year project that was co-led by Lance Cadel-Davidson. You see pictured here, and a very important person I put on as a co-author of this presentation is Beth Demings, a project manager and postdoc during the 5-year period of this project. Beth worked in my lab, but worked with all of the project team members of the VitisGen project. The title of our project was Accelerating Grape Cultivar Improvement via Phenotyping Centers and Next Generation Markers. For all the information that I don't have a chance to cover in 30, 40 minutes, please go to our website. And for the citations, publications of our project, vitisgen.org will give you further information, further background about the project. This was a 5-year project funded by the U.S. Department of Agriculture. It was a specialty crop research initiative project with an equal amount of funding from the grape industry as well as university partners that were part of this project. This was a 38 chromosome project uniting breeding programs that worked on 38 chromosome plants. We have some muscadine breeding that takes place in the southeastern U.S., and they were not part of this project, though some of the muscadine resources are a part of this project. It was a $4.5 million project in terms of federal funding that we received. Began in September 2011, which means the project just ended in August 2016. And we pulled together a collaborative project between 11 different research institutions, and we worked very closely with an industry advisory panel. A very important part of the Specialty Crop Research Initiative is to make sure that the work that we're doing is relevant to the industry and supported by the industry. Our primary objectives were fourfold in this project. There was a trade economics component to identify top priority traits, document their value. We sought to develop at least 30 new DNA markers linked with traits of viticultural and enological interests, and to apply these markers to accelerate cultivar improvement efforts. We also have an extension component, objective number 4, to get the word out to the industry, to our stakeholders. I'm going to focus my comments more on the research aspects, the genetics, genomics aspects of this project. So objectives 2 and 3, my comments will relate to. So I mentioned this was a 38 chromosome project. It included some rootstock breeding as well, so I don't just say it's a scion breeding project. But what was really interesting for me after so many years working in breeding and genetics, it's the first time I've been part of a project in which all of the breeders across the United States working on 38 chromosome plants united for a common purpose. We got everyone together from New York to California to Minnesota, South Dakota, Florida, Missouri. So these are some of the breeders, geneticists that were part of the project. Table grape breeding, wine grape breeding, and a little bit of work on rootstock breeding was relevant to this project. This project was a very coordinated effort to work in teams, to work as a team of geneticists, a team of breeders, and a team working on trait evaluation. And there were 3 trait evaluation centers that tried to do a clear, uniform job, carrying out of the trait evaluation in the areas of powdery mildew resistance, fruit quality, and low temperature stress. And low temperature stress probably relates most to climate change as some of the extremes become more common and frost damage becomes more common. But I'll be focusing a little bit more on the powdery mildew resistance and the fruit quality work done as part of this project. So these 3 trade evaluation centers would typically receive samples from members of the project and evaluate those samples, whether they were from New York or California or Missouri or Minnesota. Gavin Sachs led the Fruit Quality Center and looked at items like tannin extractability, off-odorants, organic acids, anthocyanins, and a range of other compounds related to fruit quality. Lance Cadle-Davidson led the Center on Powdery Mildew Resistance and looked at high-fold transects or visual ratings for powdery mildew resistance or colony success rates, penetration resistance. I'll say a little bit more about their work. Ann Fennell, I think many of you also know Ann for her excellent work in genomics, and she did an excellent job running the Low Temperature Stress Response Center, looking at midwinter hardiness levels, which is very important in New York and northern growing areas in the United States, but also looking at chilling fulfillment and the order of bud break and how you can genetically manipulate the timing of bud break in relation to the climate. So these phenotyping centers were there to generate phenotyping data in mapping populations. And the mapping populations, well, to work with them and to work with the phenotyping data, we of course need linkage maps. And our purpose, one of our primary goals was to relate positions on chromosomes to traits of interest in the project. When we started years ago, SSRs were the most commonly used genetic markers. Maps were not so dense. There were often gaps of 20, 30, 40 centimorgans between markers. But SSRs and some other markers were commonly used. And maps typically had 200 to 600 markers or so. What we sought to do is to develop maps with many fewer gaps, much smaller gaps, and much more of a dense map and much better coverage of the genome. So we turned to genotyping by sequencing, developed by Ed Buckler and many colleagues for use in corn, and we adapted that for application in Vitis. We worked with 2 different population structures. We worked with typical F1 populations, where we're looking at a pseudo-test cross type situation, looking for markers, SNPs that are heterozygous in one parent and homozygous in the other parent. So first we had to develop a mapping strategy, a pipeline, a software pipeline to process genotyping by sequencing data to produce high-resolution maps in these F1 populations. That was published in PLOS One last year, Jaime et al. Full reference, again, you can get on our website. For F2 populations that result from the selfing of a single F1 individual, a different type of pipeline had to be developed, and we were successful in developing such a pipeline there as well, and that was published earlier this year, and also in PLOS One, Yang et al. So the upshot of the work that we did showed that we could successfully develop maps in all the relevant populations. There were, or nearly all the relevant populations. There were, let's see if I can get, yeah, there were 17 populations. Generally, the low number was about 1,000 markers in this map, but others had up to about 9,000 markers on linkage groups. But you can see a range of different numbers of markers per linkage map, sometimes based on diversity, sometimes based on making wide crosses versus narrow crosses, large populations versus small populations. And generally, we did find that the population size, which helps us sample more meiotic recombinations, would affect the number of markers we retained in these maps. In each attempt at mapping, we'd get usually over 100,000 potential SNPs for map purposes, but there's a lot of filtering to eliminate markers that were not segregating correctly, were not of the correct type, or for which there was a lot of missing data, which is one of the problems with genotyping by sequencing data. But nevertheless, we still wound up with very useful and very dense genetic maps to use in the project. So then, now we have the trait evaluation data coming at us, the linkage maps developed, and what about the QTL analyses? Our goal was at least 30 marker trait associations, and we identified over 70 marker trait associations as part of this project. The number is still increasing as we're still processing more data. Even though the project has terminated, I think we're all thrilled with this result. There were 24 markers or 24 marker trait associations for fruit chemistry. In the area of pest and disease resistance, there were 18. 16 related to trichomes and predatory mites, and some of the trichomes provide ecological niches where predatory mites can live and survive, and some of these predatory mites control other mites or can control powdery mildew. We had six marker trait associations related to low temperature response, and 13 related to leaf morphology. Two postdocs worked feverishly in this area, John Fresnetto, now a professor at Ohio State University, and Shanshan Yang, who's moved now to another position in Arizona. Very productive pair of individuals. So I've told you a little bit about the phenotyping centers, but before I get to some of the results out of the individual phenotyping centers, let me tell you a little bit about some of the unexpected results, an area where we didn't expect so much productivity within the project, and that's this area of local phenotyping. The breeders had the funds to grow and maintain the populations, also some funds to phenotype the populations for traits of interest to them that may or may not be related to what the phenotyping centers were doing. So through the course of the project, I accumulated and tabulated the data from the various breeders. Over 100 different traits were evaluated by the individual breeding programs. They relate to foliar phylloxera resistance, botrytis rot resistance, downy mildew resistance, the never-ending study of flower sex and its inheritance, black rot, and also in the lower left corner you see cane phomopsis. It's not something you commonly see, as it's often controlled by spray programs for other diseases. But when we turn off the spray programs for powdery mildew and downy mildew, other diseases that we consider to be minor become a little bit more major. So one of the very interesting stories come out of the observations on cane phomopsis resistance. I had a technician, Steve Luce, pictured above here. Steve kept coming to me year after year for about 3 years in a row saying, Bruce, we have to phenotype for cane phomopsis. It's a very prevalent disease in certain mapping populations, but I never got to it. But Steve really noticed it. He works with the vines a lot more than I do. So one day he came back into a group meeting on a Friday morning, and he put the data on the table and showed us a bunch of slides of what these vines look like and how he rated them for cane phomopsis resistance. And right about that time, the graduate student in our lab at that time, Paula Barba, had just developed maps for those mapping populations, had just developed nice, dense GBS-based maps. She ran through the QTL analyses, and at noon she came back and said, there are two very strong QTL or major loci segregating in these populations, and this became a part of her thesis work. It's been submitted now for publication, but what's very interesting here in two related crosses is linkage group 15 lights up here in the cross of Horizon by Vitis cinerea, and linkage group 7 lights up as well in this cross. But when we replace Horizon with Chardonnay and analyzed this population for segregation of resistance. It's just the chromosome 15 locus that lights up. So we have two loci, two very strong loci for Phomopsis, cane Phomopsis resistance. And you can see this when you work with half-SIB-related populations. So a very interesting result, and we'd love for all of our QTL analyses to give us results like this, but they don't always give us results where such a high percentage of the genetic variation for the traits is controlled by the genetics. It may be a little more typical to see results like this, some work done by a student, Konstantin Divlov. And Konstantin used two different systems to rate for downy mildew resistance. He used a visual rating system developed by Atsushi Kono when Atsushi from Japan was working in our lab. And he also took pictures of the same leaf discs with his smartphone and developed a software pipeline and analysis for phenotyping for downy mildew resistance. So he could automate this and take the subjectivity out of this rating system. So he processed the photos, they become black and white, where only primarily it's sporulation that lights up. He counts numbers of white pixels, and in both cases, he could come up with the same QTLs, but this takes a lot less time. This is a much easier assay to do based on computer vision. So we're developing technology, but what came out of this work were a number of different linkage groups that we found were involved in downy mildew resistance, from Vitis rupestris and from a cultivar, a Saval-descended cultivar called Horizon. And, but the percent variance explained is rather low, but altogether, they do explain a good percentage of the variance for downy mildew resistance. So a nice project coming out of VitisGen here. Okay, so let's talk a little bit about powdery mildew and some activities undertaken by the powdery mildew trait evaluation team. This is Lance Cadel-Davidson, David Gadori, and a good group of technical staff and all of us breeders sending material to them. So they were very interested in precise phenotyping, precision phenotyping approaches. And going into this project, the standard was to use for the inoculation with a single strain and quantify resistance by looking at penetration success and hyphal growth. So do you get naprosorium to form, and do you get a colony to begin to grow? But we quickly learned that this was not the best approach to quantify QTLs or genes with smaller effects. So we tested the plan A versus plan B. Let's look at the amount of hyphal growth with a known amount of inoculum, and let's rate this 8 to 9 days post-inoculation and rate this based on hyphal transects. How many times does a hyphae cross a particular line in a field of view? So they settled on doing further work at the point that they did these experiments. Further work was then done using hyphal transect data. And now they also took on the question of vineyard approaches versus laboratory approaches. Breeders typically rate in the vineyard where conditions are variable. The epidemic may vary from one location to another. The strains may vary from year to year, or from one end of the field to another end of the field. So in some data, this is data based on REN1 in California. With field rating, sometimes you can detect REN1 on linkage group 13, sometimes not. So there are 3 ratings shown here on the left. We don't always detect this REN1 locus. But if you look on the right side, 4 different rating experiments, a much higher percent of the genetic variance, the R-squared values are much higher. It's all mapping as expected to linkage group 13, but very consistently, 2012, 2013, 2014. So it was a nice experiment to do, which gave us a little more confidence in the ratings that we were doing in the laboratory. So we settled on a precision phenotyping approach in the laboratory where breeders send samples, they're processed, they're sterilized, they're inoculated with single strains, they're placed into what amount to cooking trays, glass cooking trays with a layer of agar. They're incubated under very uniform conditions. You have an opportunity to do a great deal of replication with these experiments, but they do take time to count. But you can store the material and count them over a period of months, count your hyphal transects. So that was done for a number of populations, and the Phenotyping Center also looked at specificity and durability. I know there's going to be more said about this a little bit later, so just briefly, in a series of papers in which they examined plants with combinations of run 1 and other loci stacked in a stacked situation, the question we were trying to answer was, which loci can we use to help protect run 1? We know that there are powdery mildew strains that overcome run 1 from the muscadine grape, especially strains collected from southeastern U.S. that co-evolved with run 1. The answer in the series of papers was Ren 2 may offer some protection, Ren 2 from Vitis cinerea, and possibly the rupestra source would offer some protection as well. Ren 4 is very interesting in itself in that so far with strains that we've tested, we do not see that Ren 4 can be readily overcome. It's not so race specific unless we find with further testing that Ren 4 from Vitis romanettii can be overcome. But it also appears to have the value of being able to protect Run 1. Finally, out of the Powdery Mildew Center, one of their main goals was to document other sources of resistance, and this is an example out of the University of Minnesota. I'm not going to spend much time due to the amount of time I have in trying to explain this, but the group used 2 half-sib families descended from Minnesota 1264, and did a little haplotype analysis of a linkage block descended from linkage group 2 and one from linkage group 15. And in 2014 and 2015, these were the most resistant ones, having both blocks from linkage group 2 and 15. The chromosome 15 source is Ren 3, based on the position that we could narrow this down to. But the chromosome 2 source was novel and recently named Ren 10. The paper is in press at this point. We're hoping to hear soon about a DOI number from molecular breeding. Now let's summarize some of the results from the Fruit Quality Center, and I emphasize the positive and negative aspects of fruit quality. As a breeder who works in making crosses, interspecific crosses with wild species, I really want to eliminate the off characteristics that come from these wild species. So that's really is an issue, not just high quality, but the negative aspects of quality that are known to exist in wild species. They could be green, herbaceous, stemmy aromas. And in this area, we did find some putative QTLs for the C6 aldehydes like hexanol and hexanal. We found QTLs also related to anthocyanin production and the types of anthocyanins, juice protein, sugars, yeast assimilable nitrogen. Some of this has been published in the publications listed at the bottom of this slide. But let me give you the one example from the fruit chemistry, fruit quality section on the inheritance of excessive malate, which typically comes from Riparia or Cinerea, some of the species that are extremely high in malic acid production. In vinifera, the typical titratable acidity level might be 5, 7, 8 grams per liter. but in wild vitis, this can be 15 to 40 grams per liter. In a mapping population, we saw a great deal of segregation. I believe this was the Riparia bisaval F2 population. There was a great deal of segregation for level of malate. We ran GBS and QTL analysis, and there's a very strong association, a significant association that pops up on linkage group for aluminum activated malate transporter enzyme. And the gene is also associated with malate accumulation in apple and some other species as well. So it's a potential candidate gene at this location. We haven't proven that this is the gene responsible for malate from wild species. There's more work to do, but it was very interesting to us to find in this interval on linkage group 6 that ALMT exists within the confidence interval. The trait evaluation work was successful, a range of markers produced in the project. The breeders were busy every year in this project trying to apply what was known already about marker-assisted selection possibilities, and later in the project trying to apply what we had learned during the project to marker-assisted selection. In the early period, we tried to validate SSRs and improve our SSR selection techniques. And we focused quite a lot on markers for flower sex, seedlessness, powdery mildew resistance, downy mildew resistance. A few others were tried out, but I'd say the bulk of the successful work was in the area of disease resistance. And let me give you examples from my program where we use marker-assisted selection over the course of 4 years on about 15,000 seedlings total. And over the period of time, the percent that we retained decreased as we began to stack more and more markers together. And the number of seedlings represent multiple populations each year. There was some additional phenotypic selection that would take place in the nursery each year. But in this most recent season, we took 3,700 or so seedlings that were sent off for DNA and marker analysis, processed the data. Only about 25% were planted out to the field, and of that 25%, we reduced another 10%, so 15% altogether remained at the end of the season after phenotypic selection in the nursery, which was preceded by the marker-assisted selection. Did it work? Yes, it worked. We did validation with REN1, RUN1, other markers, and when we used these markers en masse, the nurseries looked extremely clean, powdery mildew-free, and almost downy mildew-free under no spray conditions year after year. This year, of the 900 or so vines that were planted, I think 4 or so were misclassified, and I think we found 4 covered with powdery mildew. Not too bad if 896 were good and 4 were not. Very good success rate. We also, as part of our project, we had some unexpected benefits. We had the challenge of trying to develop new markers, starting with SNPs based on genotyping by sequencing. So you could either take the approach of looking for nearby SSR markers that were polymorphic, or develop a new approach with next-generation sequencing technology. And we took that approach to develop this idea we call amplicon sequencing, where many different primer combinations can be multiplexed, and many different seedlings can be run with the same multiplex and with barcoding through the same sequencing lanes on a MiSeq sequencer. And you could either look for the presence of particular tags. Do they amplify? Does the tag for RUN1 amplify? Or does the tag for REN4 amplify? Yes or no? And yes here, no there. Or you can look at the haploblocks surrounding a locus of interest. This is the REN2 haploblock. Is it present or not? This is the genotype expected if REN2 is not present. And in orange, these are the resistant types generally. Resistant seedlings here. And we could use this simultaneously in one step to stack genes too. If we make a cross that incorporates RUN1, REN2, REN4 as well, we could use this approach to stack our genes. So it's a new technology developed within the project, and we're very happy with the development of AMP-seq. So we're at a stage now where the breeding programs are beginning to have the seedlings from the early stages of marker-assisted selection bear fruit, and these were a couple of examples of crosses made in 2012 that looked like this just about a month ago in our vineyards. 6 weeks ago, I'd say. 2 crosses made in 2012, both white wine grape types, reasonable sized clusters. The one on the left, a fairly loose cluster type. They taste good to me. I know nothing yet about the wine quality, but I see these highly resistant types grown under no spray conditions, highly powdery and downy mildew resistant, and the one on the right, the red one, has a little more perfumey fruitiness to it. So I think they have good potential. Of course, time will tell. As breeders know, these may not be the ones that would ever be released, but they're at the early stage in the program. In addition, we were able to genotype older selections within the program, and this one we do have some wine quality data for. We know it's stacked with RUN1, REN2 for powdery mildew resistance, RPV1 for downy resistance, and excellent resistance to downy and powdery. We know it's quite resistant to black rot, as we had a good test year for that last year. Very good winter hardiness down to minus 26°C, maybe not necessary in this area of the world, but in many areas of the world, yes, minus 26 would be very good resistance to have to bud kill in midwinter. And some wine quality indications, at least 2 wine samples already indicating some nice blackberry, plum, cherry, green pepper, some tannin, and some interesting chocolate notes to this one too. So to summarize, we've developed a pipeline for sample processing and data analysis with both GBS and SSR markers. We have a library of over 2.6 million SNPs that were identified using diverse collections descended from multiple species, some Asiatic species, some North American species, 16,000 plus vines already analyzed in the project. We have medium to high resolution genetic maps developed in 16 of our core families, more than 70 marker trait associations. We have given breeders the tools and the funding within this project to carry out marker-assisted selection and carry out more activities in precision breeding, and the tools to stack genes, to stack multiple loci for powdery mildew resistance, for example. And the AMP-seq technology, I think, will have multiple uses even beyond marker-assisted selection. There's a great deal of interest that's been generated in this area, and we're hoping to eventually be able to develop a series of new cultivars for the future. But through the course of this project, we feel we've put a lot of information and a lot of tools into the hands of grape breeders. So the team, many people to thank, too many to name and to mention, but also our funding sources, USDA, National Grape and Wine Initiative, Table Grapes of California, Gallo Winery, Cornell University, and all the other universities associated did their part in funding this project. And not to be missed are our advisory panel and a large group of students, postdocs, technicians that were associated with the project and helped us get to where we've gotten to within this project. So for me, I went in with some doubts how to coordinate such a large project, and now I'm anxious to do it again, and we're in the process. I think this was very successful, and we're in the process of trying to reapply and continue the project. There's much more we'd like to do. So with that, I think I wrap up, and I thank you very much for the chance to speak to you today. Thank you.